Hello students, the topic of today's lecture is glyoxalate cycle, a variation of the tricarboxylic acid cycle or Krebs cycle. The glyoxalate cycle, an anabolic pathway, centers on the conversion of acetyl coenzyme A to succinate for the synthesis of carbohydrates. In plants, certain invertebrates, and some microorganisms, including E. coli and yeast, acetate can serve both as energy-rich fuel and as a source of phosphophenol pyruvate for carbohydrate synthesis. In these organisms, enzymes of the glyoxalate cycle catalyze the net conversion of acetate to succinate or other 4-carbon intermediates of the citric acid cycle. You can see in the glyoxalate cycle, acetyl coenzyme A condenses with oxaloacetate to form citrate, and citrate is converted to isocitrate exactly as in the citric acid cycle. The next step, however, is not the breakdown of isocitrate by isocitrate dehydrogenase, but the cleavage of isocitrate by isocitrate lyase, forming succinate and glyoxalate. The glyoxalate then condenses with a second molecule of acetyl coenzyme A to yield malate in a reaction catalyzed by malate synthase. The malate is subsequently oxidized to oxaloacetate, which can condense with another molecule of acetyl coenzyme A to start another turn of the cycle. Each turn of the glyoxalate cycle consumes 2 molecules of acetyl coenzyme A and produces 1 molecule of succinate, which is then available for biosynthetic purposes. The succinate may be converted through fumarate and malate into oxaloacetate, which can then be converted to phosphoenol pyruvate by PEP carboxykinase and thus to glucose by gluconeogenesis. Vertebrates do not have the enzymes specific to the glyoxalate cycle, that's isocitrate lyase and malate synthase, and therefore cannot bring about the net synthesis of glucose from lipids. In plants, the enzymes of glyoxalate cycle are sequestered in membrane-bounded organelles called glyoxysomes, which are specialized peroxisomes. Those enzymes which are common to glyoxalate cycle and citric acid cycle have 2 isozymes, one specific to mitochondria, the other to glyoxysomes. Glyoxysomes are not present in all plant tissues at all times. They develop in lipid-rich seeds during germination. Before the developing plant acquires the ability to make glucose by photosynthesis. This is all about glyoxalate cycle. Thank you.